Hello, and welcome to the Scope of Things podcast, a no-nonsense look at the promise and problems of clinical research based on a sweep of the latest news and emerging trends in the field, and what I think is worthy of your 30 or so minutes of time. I'm Deborah Borfit, Senior Science Writer for Clinical Research News, which means I spend a lot of time with my ear to the ground on your behalf, and a lot of hours every week speaking to top experts from around the world. Please consider making this your trusted go-to channel for staying current on things that matter, whether they give us hope or a cause for pause. In another 10 minutes or so, I'll be speaking with Erin Mackey, Vice President of AI and Data Science at Lookavant, about correlation versus causation in clinical research. But first, the latest news, including the pending launch of a large treatment trial for Graves' disease, growing popularity of quality of life measurement in cancer clinical trials, a digital-first recruitment campaign for a diagnostic tampon, a reference trial emulation study on a pair of blood thinners, a new strategic partnership for Walgreens, a detective algorithm that could accelerate the design of certain trials, and how eligibility criteria has been inappropriately excluding people of African or Middle Eastern descent from cancer studies. Before the year ends, Immunovant will be launching a late-stage clinical trial for Graves' disease, the first to happen in a very long time. The treatment is known as IMVT-1402, a re-engineered version of the company's first-generation asset known as batoclimab. The therapeutic approach targets the neonatal Fc receptor, most famous for transferring antibodies from mom to fetus during pregnancy, but that in Graves' patients also recycles harmful immunoglobulin G autoantibodies that in turn stimulate excess thyroid hormone production. If successful, the second-generation 1402 treatment would bring welcome relief to the roughly 1/4 of patients who are hard to treat with methimazole, approved for medical use in the U.S. since 1950. The most recent Phase 2A trial found that in patients uncontrolled on anti-thyroid drugs at week 12, high-dose batoclimab achieved a 76% response rate in combination with methimazole and 56% on its own. And a strong correlation was seen between degree of IgG lowering and clinical outcomes. In addition to Graves' disease, at least 10 other conditions could be targeted with anti-Fc receptor medicines, collectively representing about 800,000 patients in the U.S. alone. You can read all about it in a story that was recently published in Clinical Research News. Expect quality of life measures to become commonplace in cancer clinical trials, thanks to the development of new measurement systems allowing such metrics to be effectively and precisely assessed. In a first-ever nationwide trial looking at the quality of life trajectory of young Hodgkin lymphoma patients, the addition of an immuno-oncologic agent to a multi-drug chemotherapy backbone proved to have fewer negative side effects on their quality of life than standard treatment, and was not just superior based on standard clinical measures like overall survival. The team behind the study is now collaborating with several other groups in discussions with the National Cancer Institute and the U.S. Food and Drug Administration about how to incorporate the patient experience into the drug approval process across the entire age continuum. As was aptly pointed out by one of the investigators I spoke with, quote, "We now have the luxury to care about quality of life for certain types of cancers that have very high survival rates," unquote. The inclusion of these metrics in clinical trials means patients better know what to expect of treatment and enables a comparison of choices based on outcomes as defined by individuals with the same condition. In the show notes, you'll find a link to the full story. Women's Health Startup Day credits a partnership with Lindus Health for amping up recruitment for a clinical trial of a diagnostic tampon for the self-collection of vaginal and cervical fluids analyzed for biomarkers of sexually transmitted infections. A digital-first recruitment campaign to help bring in 500 participants in the first week, expediting the product's path to market in the U.S. and Europe and aiding the cost-minded company in its mission to bridge the gender diagnostic gap that has left many women fed up with the current standard of care. The tampon is a familiar device used by over 90% of the female population and much better suited to their anatomy than conventional vaginal swabs, as was shared during a session co-presented by Day and Lindus at the recent Next Generation DX Summit. The trial, which enrolled diverse participants between the ages of 25 and 65, found not only that the women preferred the diagnostic tampon over the swab, but outcomes compared favorably to a variety of other self-screening methods, as well as a clinician-administered program. A full write-up on the presentation can be found in both Clinical Research News and Diagnostic World News. In the UK, researchers recently emulated a large randomized controlled trial testing the effectiveness of two blood thinners using data from real patients to explore treatment effects in people underrepresented in the study for safety or feasibility reasons. Patient eligibility, selection, and analysis approaches in the reference trial simulation mirrored that of the original Aristotle trial. However, results differed somewhat between the two, which was attributed to, among other things, the fact that the real-world cohort had fewer patients of Asian ethnicity known to be at a higher risk of stroke when taking the older oral anticoagulant drug and greater overall use of concomitant medications. Plus, a bias was introduced by patients who started on one drug but switched to the other. Studies like these are helping regulators better understand the potential and limitations of real-world data. I spoke with a lead researcher on the UK reference trial emulation study, and the resulting story can be found by visiting clinicalresearchnewsonline.com. Walgreens and the Biomedical Advanced Research and Development Authority, or BARDA as it's better known, recently announced a strategic partnership around a decentralized clinical operations for healthcare and research program, one of whose goals is to enhance clinical innovation for more efficient and relevant research. The first of its kind partnership will utilize Walgreens clinical trials and BARDA's expertise to address barriers in decentralized clinical trial access and conducts trials over a 5-year period. BARDA's plan is to leverage the partnership to expand trial access to diverse populations and help with future public health emergencies via decentralized clinical trial settings, to include Walgreens community pharmacies. I'll link to a news brief on this in the show notes. Researchers in Spain have built a detective algorithm, a publicly available resource called RT Detective, which predicts the best drugs for treating diseases caused by mutations that can bring protein synthesis to a halt, which could accelerate the design of clinical trials for certain genetic disorders and cancer. This marks an important step in helping personalize treatment by matching patients with specific mutations to the most promising drug candidate. A recent study suggests that trials to date using so-called nonsense suppression therapies could have been more effectively used if the genetic code immediately surrounding the nonsense mutation, and not just the mutation itself, had been considered. The discovery came after studying nearly 6,000 premature stop signs, as they're called, causing disease and testing the efficacy of 8 different drugs on each of them. RT Detective was used to predict the effectiveness of drugs for each of the 32.7 million possible stop signs that can be generated in RNA transcripts in the human genome, achieving higher read-through percentages that generally correlate with better therapeutic outcomes. A link to the study published in Nature Genetics can be found in the show notes. And finally, diversity in cancer clinical trials could get a boost with one simple move, namely modifying the threshold for neutrophil levels in study eligibility criteria. In people of African or Middle Eastern descent, the so-called Duffy null phenotype is a predominant trait that results in relatively lower levels of the white blood cells when measured in the blood. This is not because they have less neutrophils overall, but because they are more frequently located in other body tissues. People with adult Duffy null phenotype are equally able to fight off infections compared to others, yet more than 3/4 of them were inappropriately excluded from 289 major phase 3 trials, researchers have found. The additional problem is that treatment guidelines on dosing are based on the trials in which those drugs were tested. Click the link in the show notes for all the study particulars. It is now time for a Q&A with Lokavant's Aaron Mackey to explore some of the unintended consequences of decisions made during trial planning and execution that can lead to questionable conclusions. Welcome to the show, Aaron. I can't wait to get started on this topic. Ready to roll?
Hey, Deb, thanks for having me on the scope of things. I'm really excited to talk about clinical trials and causal AI.
Excellent, excellent. So happy you're here. The overarching issue, I believe, is confusion over when study variables are causative versus when they are simply correlated. Since these are related concepts in research that can be difficult to distinguish, Aaron, can you give us a for instance that you have seen come up again and again when study teams are dialoguing about a planned clinical trial or tracking progress once it is launched?
Sure, thanks. So, during clinical trials, we perform a lot of so-called conduct analytics. We measure KPIs and QTLs and lots of other acronyms, metrics that are supposed to help us understand what's going well and what isn't. And when one of those numbers goes off the rails, so to speak, we want to know, okay, great or not so great, how do we fix it? What should we do to change the situation to improve that metric? So one of the things that's often done is we then go to historic observational data, what happened in the past, and we try to make associations between those situations when the numbers looked good and those situations when those same numbers did not look good, and ask, well, what was the difference?
Mhm.
And of all those very many differences, there may be some things that we say like, oh, well, that difference is something we can act on and try to make one of those bad numbers look good. So let me take patient recruitment or patient enrollment as a more concrete example. So we're running a trial, we're seeing lower recruitment rates than expected, and we have various options that we want to consider to improve it. Should we add more countries? Should we add more sites? Is there a change? Is there a problem with the eligibility criteria? Is there an aspect of the eligibility criteria that is perhaps too restrictive that we could consider loosening? Again, we may go back to some historic enrollment rates for similar trials and look at differences in eligibility criteria, differences in country site mixes. And based on this, we might be led to make a change, a protocol amendment even to that eligibility criteria. But we have to ask ourselves, what guarantees do we have that if we make that change, that that's going to actually improve the metric, that's in this case is going to actually improve the enrollment rate? Because
Mm.
maybe those historically faster trials that did happen to have looser criteria, maybe they also had some larger or different patient outreach campaign or a stronger site to patient network engagement or some other factor that actually had nothing to do with the eligibility criteria, but just happened to be a so-called hidden confounder that sour an otherwise sensible sounding approach.
Mhm. Easy to go wrong for sure. Just so how sideways can things go, considering everything that goes into trial design and execution? Decisions about, as you talked about, patient eligibility and then site selection and how data will be collected and even what the trial endpoint should be. Have you more often observed that problems are perennial and tend to pile up, or are these more easily fixable one-offs specific to a trial and study sponsor?
It's a mix of both, of
Okay.
course, as you would expect. So, in some previous research in my group, we found, perhaps not too surprisingly, that there are clear institutional biases, particularly among large pharma sponsors, evidence of what I call protocol copy and paste sort of templating problems, where each new trial starts with a trial protocol that is essentially some reincarnation of past history, of past sins, even. So those
Mhm.
can continue to remain unaddressed and systematic and repeated over time. In fact, that same team built a particular type of statistical classifier that could predict which particular pharma company authored an otherwise anonymous eligibility criteria block of text with about 80% probability of getting it right. So it's just a sort of example of how, even though you may look at it at a particular part of the protocol and say this looks fairly generic, there are these sort of historical sort of traces that happen. But that said, a lot of one-off problems occur, particularly as trials get more and more specialized and more specific to, increasingly narrower and even rarer patient populations. Even among what would otherwise be considered common diseases, many of the trials have become, again, through very narrow eligibility criteria, addressing rarer and rarer patient populations, where in fact now the historic record may be fairly thin, or even the indication is still relatively novel. And there's a sort of old quip that, it's always hard to make predictions, and particularly about the future.
Mhm, mhm. Let's talk about the real and invisible costs when assumptions are made instead of relying on cold, hard facts, and where causal insights have the most to offer study sponsors in terms of truncating timelines and shrinking budgets, which I know Logovan likes to talk about a lot.
Exactly. Yeah. So I think a lot about timelines and budgets at Locovant. So thank you for asking about that. Specifically, so for us, right? So you can, in some ways, you can always trade time for cost, and vice versa. So if I want my trial to enroll faster, well, then I can open up more sites, and that will cost more money, particularly in the site opening and site engagement. But I will get to my target enrollment faster, all other things being equal, and vice versa. If I want to save money, I can open fewer sites, and now my enrollment may take much longer, but I will achieve it at that lower cost. And so there's this sort of fundamental trade-off there. But to answer the question, like, okay, well, how do I hit my sweet spot? How do I optimize then for a particular budget or for a particular time constraint? What's my causal expectation? Again, we sort of keep harping on this term of of, right, there's historic data about how many sites gives you how many patients in those trials, but can I really expect that from my trial? How can I account for those possible, again, those confounders, either confounders that I know about? I expect that recruitment is going to be different region to region, site by site. I know that recruitment may be different in different countries or different areas. So how can I both account for both what I know should be different and then also all of the ones that all of the sort of hidden confounders. So what's needed and what I think the industry as a whole is turning towards is more not just predictive analytics, but more prescriptive analytics. Algorithms and machine learning, AI even that makes recommendations about what to do next. And do here is really the key causal word here, right? The Judea Pearl many years ago came up with the so-called do operator. It's the perturbation, it's the lever that you can pull, it's the knob you can turn. That having done that, having pulled that lever, what is then the expected effect of that action? And not just the expected effect on average overall, but for your specific trial, having adjusted and accounted for all the particular specificities of your trial to get a kind of individualized effect. That's really the key difference that causal AI makes for a sponsor or a CRO who's looking to improve upon the ordinary.
Are you enjoying the conversation? We'd love to hear from you. Please subscribe to the podcast and give us a rating. It helps other people find and join the conversation. If you've got speaker or topic ideas, we'd love to hear those too. You can send them in a podcast review.
I'm glad you brought up AI because I wanted to ask you about that in a broader sense, actually. Do you personally use like ChatGPT or any other large language model? And if so, for what reasons? And I want you thereafter to sort of follow up on that with how you're using AI and machine learning more specifically at Lokavant and your work there, and maybe some kind of comparison of those experiences in those two different realms. And I don't know, and if these experiences will make you more or less, skeptical of the output.
Sure. So I would say as a practitioner in the field of, data science and computational sciences in the pharmaceutical industry for the last 25 years, I've been using computational tools that make my work easier all the time. Going back to even in, the Microsoft products within Word or PowerPoint, taking suggestions about my grammar, my spelling, my this or that. Right. So in some ways, while we talk about these tools as being brand new, they've actually been around for a very long time. And people who are good at their craft have learned how to embrace them, have learned how to use those tools to make their work faster and make their work more efficient. So, certainly, myself and my team members, I encourage everyone, if you haven't already learned how to use these tools to make your work easier, again, whether that's generating text, whether that's generating slides, whether that's generating images, whether that's generating code. And in fact, most of our work is in data science and AI does revolve around authoring code to answer questions. To not use those tools is to really have, one, if not two of your hands tied behind your back. So certainly embracing these things. Now, that said, there still has to be a human in the loop. Those tools are not doing the work for us. They're assisting us. They're driving us. And in particular, one of the biggest gaps with the tools and something that sort of a transition to our work at Lokavant is that they don't really have a lot of very specific domain knowledge. If you ask ChatGPT about clinical trials, it will give you some generic sounding, Wikipedia type answers about trials in general. Mm-hmm. But if you ask it about a very specific trial or a very specific treatment or a company, it doesn't know anything about that. It doesn't have specific information. So you have to then build information systems that support them using things like knowledge graphs or other sort of back-end databases that the tools can then use to put together so-called chains of inference, where when you ask a question or a series of questions, there's a particular set of steps that the language model will go through to try to answer the question. And we again, myself and teams that I've worked with have built these systems where they're really kind of expert knowledge systems in a particular domain. So what we're doing at Lokavant is taking that concept and saying, well, in our domain, what are the particular things that a language model needs to be retrained on? And it's things like eligibility criteria, protocol text, site and patient level, demographics and geographics and sort of geospatial distributions. Those are the sorts of domain information that we can essentially feed to these models now, and then ask questions about how do these particular differences in protocol text become then associated or even causal, have again that chain of causal inference to make to deliver insights and even recommendations.
Wow, very good. Yeah, I know that Lokovan has been in the news a bit advocating for the use of AI to improve clinical trial diversity, as is now mandated by the FDA, and has made the point that selecting study sites and investigators in the conventional way based on previous enrollment performance, kind of what you're pointing to a moment ago, won't help study sponsors meet the mark. Can you share with listeners about what the new data feeds need to include to ensure the right proportion of different population groups, make sure they're participating in their trials?
Sure. So it's a really important question. And, the not so dirty little secret about that so-called conventional method of, looking at previous enrollment performance, I think we're understanding as an industry more and more that's not actually a very reliable method. And once you actually control for the sort of knowable differences in the different trial operations, including how much site training there was, how much CRO or sponsor site engagement there was, what tooling was put together, what the incentives were. Once you kind of account for those differences, that historic performance is not a very good predictor of future performance overall. But yet it's something we continue to use. It's still, I think, as you say, a sort of industry standard, and there's a bunch of biases associated with it. So I'm really glad you asked about diversity, because in clinical trials, it's really key to ensuring that results are not only interpretable from a scientific perspective, but that when we now go into regulatory and even sort of payer negotiations, we need to understand, are the results applicable or even generalizable to a wider population of parents? And this is not just as a practitioner in the field, but as a sort of taxpayer. I want to know that when the regulatory agents are approving something, that they actually have the data to say that this is going to be a benefit to patients regardless of their ethnicity, regardless of their socioeconomic status, regardless of where they live and what their access to healthcare is. So as you say, access to clinical trials has always been biased towards population centers. Of course, we always see that the most highly performant sites are the sites where there are the most people. If you want to enroll your trial as quickly as possible, put all your sites in Manhattan. I like to joke, and in fact, that would work if there weren't already so many clinical trials already running in Manhattan that it's essentially entirely saturated.
Mm-hmm.
So there is a competitive aspect there. So these that bias towards population centers that has their own socioeconomic and even political biases, that leaves a very systemic ripple effect that that ripples out into the equitable or essentially non-equitable delivery of healthcare across the U.S. and even the whole world. So our methods for site selection, to answer your question, the methods for site selection and optimization have to begin accounting for these biases, kind of looking at them up front, using real-world data sources to say, okay, great, my patient populations, yes, they're in Manhattan, but they're also in Dubuque, Iowa. And even though I don't have very many sites in Dubuque, Iowa, I can put some there, and I'm going to capture a different patient representation there than I would otherwise. So we have to optimize not just for enrollment and operational costs, but other objectives, including those diversity objectives. And your news item about the Duffy null phenotype is a great example where again, it's not just the enrollment rates, it's the overall protocol design itself, going back to what is the eligibility criteria? What is the patient burden? How, what does that sort of site visit, site complexity look like? How do all those things bias who can actually participate in the trial or not?
Yeah. It certainly helps to have AI, as your right-hand man, so to speak. But my last question is going to go in a total different direction. I just want to have a little fun here, Erin, if you'll allow me. I want to sort of give a nod to all the tech-weary people out there, which no doubt includes all of us from time to time. If we were all on board for a prolonged tech holiday, or cyber thieves and wayward satellites were to prevent us from accessing the internet and all our connected digital gadgets for we know not how long, What would be your stopgap emergency plan for keeping ongoing clinical trials on track with no digital data or AI at your disposal?
Wow. Well, I hope we're not going to go back to using an abacus to sum up our adverse event counts. But I do think your question points to, to one aspect of clinical trials that deserves a lot more attention, which is, we talk about all the electronic ways to capture data and electronic ways to transmit data and electronic ways to kind of monitor data and automatically identify things and have dashboards and alerts and send information to patients through their digital devices and collect information through their digital devices. And that's just, screams of, efficiency and data transfer, and it sounds just wonderful. But I think the part that all of that misses, and I think to your question, is there's a human-to-human interactive aspect of a patient participant who is participating in a clinical trial, and they're looking to get something out of it. They're doing it not just for fun. Usually, they're looking for some reward. Maybe it's some aspect to their own personal health, some other philanthropic desire to contribute to pharmaceutical research. There's something there that they're looking for, and being a part of that trial and having some interactivity with the humans that are involved in that trial, I think is really important. And it's something we often kind of overlook when we're focused too much on the sort of digital connectivity of all the various people involved. So I think the site investigator, the site coordinator, all of the sort of site staff that actually make the trial happen, and that connection with the patient and their own healthcare people, remain, on that deserted island where we don't have access to that. That's what we would be left with. And yes, we may be left with scratching on clay tablets to do paper CRFs, but I think we would recapture in some way some of that human element. And I've said it already, but I really am a strong believer in that the best AI is still human in the loop AI, where the various digitalization is there to help us and not to get in the way. So I don't believe that AI is ever going to truly replace humans, but it can help us work better, work faster, and improve and make easier the things that we're trying to achieve.
Yeah, well, hopefully nothing so nefarious as a cyber attack or wayward satellites would ever come to pass, but maybe a brief holiday might bring that point home. Things would at least get interesting pretty fast. Anyway, I thank you for keeping it real when it comes to what technology can do for clinical trials, as well as the room we need to leave for common human sense. This was fun. Thanks again for agreeing to be my guest on today's show.
Deb, thank you so much. This is great. Happy to come back anytime.
Well, you will be invited back for sure. As always, a big thank you for everyone out there listening in. If you're not subscribed to this podcast yet, please consider going to Apple Podcasts and doing so right now so you don't miss your monthly dose of news and perspectives you'll be hard-pressed to find anywhere else. And if you're up for it, I'd be so very grateful if you'd leave a rating and review while you're there. One more thing before we go, if you like today's conversation, it is only a glimpse of what you can expect from Scope Europe presenters and panelists. Please plan to join us October 29th and 30th in Barcelona, Spain, when clinical operations executives will be sharing best practices and discussing optimizing digital and hybrid trials in Europe and across the globe. Save an additional 10% off any current rate by using the code SOT10. For more information, visit scopesummiteurope.com. Bye for now.